This is Matt Dean with A+ College Ready, and today we're going to talk about enzymes. Enzymes are catalysts. They're proteins that help to increase the rate of a chemical reaction without actually taking part in the reaction. Enzymes aren't changed by the reaction that they help to catalyze. They don't get used up. One of the most important things about enzymes is that they decrease the activation energy of a reaction. That's the amount of energy needed to get it started. With enzymes, reactions can happen with less activation energy than without them. As we said earlier, enzymes are proteins. They have a three-dimensional shape, and that shape is very important to their function. An important theme in biology is that the structure determines the function. This is true for enzymes. This shape makes enzymes very specific. Each enzyme can only catalyze one particular reaction. You have many different kinds of enzymes in your body. The shape of an enzyme is maintained by both hydrogen bonds and disulfide bonds, just as the shape of any protein is maintained. The substrate is the substance or substances acted upon by an enzyme, whereas the active site is a groove in the enzyme where that substrate will bind. The substrate and the active site fit together sort of like a key fits into a lock. And again, this is one of the main reasons that enzymes are so specific and that they only catalyze one particular reaction. In addition to the shape being important, the charges and the amino acids found at the active site also help to determine what can bind there. This graph shows you two different pictures for the same reaction. The one on the left has no enzyme present. The one on the right has enzyme. So notice the amount of energy present in the reactants starts out here, starts out at the same place for both reactions. The amount of energy in the products ends up at the same place. So this reaction is an exothermic reaction. It's releasing energy to the environment. Notice the only difference between the two is the amount of energy needed to get this reaction started, the activation energy. With enzymes, it takes much less activation energy than without. All right, so here's an example question. We have a graph on the left, and we want to know, is the reaction illustrated in the graph endothermic, which means does it release energy to the environment? I said that backwards. Does it absorb energy into the environment? Or exothermic, does it release energy to the environment, and why? Well, the key is to look at how much energy the reactants start with and how much energy the products end up with. And you notice that the reactant started with less energy than the products ended up with. What that's telling you is that the reaction is absorbing energy from the environment. And that means that it's an endothermic reaction. So our answer on this example is B. The endothermic reaction is absorbing energy. The products end up with more energy than the reactants. Also notice the amount of activation energy. To get this reaction started, we need to add an amount of energy from here to here. So that amount of energy gets the reaction started. And that's the activation energy again. All right, so this slide sort of symbolizes or illustrates how enzymes work. So here's an enzyme. Notice the groove with a very specific shape. That's the active site. Here's the substrate. Notice that the shape of the substrate and the shape of the active site are complementary. So the substrate fits in. When they're together like that, we call that the enzyme-substrate complex. Once they bind together, the shape of the enzyme will change a bit in a process called an induced fit. You might also hear it called a conformational change. And that little change in the shape of the protein, the enzyme, will help to catalyze the reaction. In this case, it's probably going to pull apart the substrate. It's going to pull on its edges, and it's going to break a bind. So we might say that this represents a degradation, or we might say a catabolic process, a breakdown process. Notice that when the reaction's over, the enzyme is not changed. It's exactly the way it was to begin with, but the substrates have turned into products. Sometimes you'll see this written as enzyme plus substrate yields enzyme substrate complex, which eventually yields an enzyme unchanged plus the products. Most people have a misconception about enzymes that they all cause breakdown. But notice on the right, here's an enzyme. Two different substrates will fit into that enzyme, into its active site. A conformational change will take place during that enzyme substrate complex. And in this case, the substrates are combined to make a product. So enzymes can cause catabolic processes to happen or degradation processes, but they can also cause synthesis or anabolic buildup processes. So we said before that enzymes lower the activation energy. Probably the main reason or the main way they do that is through substrate orientation. They help to line up substrates so that they collide with the proper orientation to react. Without enzymes, substrates may bump into each other, but if they don't bump with the right orientation, they won't react. Without an enzyme, about the only way to make that reaction happen faster is to heat it up. Because remember that heating causes materials to move faster. So if they're moving faster, they're more likely to bump. And the more times they bump, the more likely they are to bump with the right orientation. So heat can be a form of activation energy. Enzymes lower the amount of heat needed because they automatically line up to substrates. Enzymes can also put physical stress on a bind. So when a substrate binds into that active site and that induced fit happens, it can pull on the bind and cause it to break. They can also cause changes in substrate reactivity. They can help things like a phosphorylation or a redox reaction take place. So here's an example question. During which time interval is the rate of the reaction the greatest? Notice that we're looking on the y-axis at the number of moles of products formed, and this is during an enzyme reaction, graphed against time. The rate of an enzyme catalyzed reaction is always going to be the fastest at the beginning. And the reason for that is because at the beginning there are more substrates. Over time, the substrates are converted to products because that's what the enzyme does. It converts substrates to products. So when there are less substrates, it's harder for enzymes to find them. They're less likely to bump into each other. They're less likely to collide. So the answer we're looking for here is 0 to 10 seconds. You can also mathematically look at that by taking the slope of the line between each of these time intervals. And you'll find that the slope from 0 to 10 seconds is higher than the slope between 10 and 20, also between 40 and 50. The rate is definitely not constant. If it was, you would get a nice straight line. Here you get a curve. When you get a curve, that means the slope is changing. It also means the rate is changing. So here we want to find the rate between 10 and 20 seconds. So we're looking at the rate in that particular part of the graph. Remember that when we have a graph to find rate, we want to find slope. So we want to find the coordinates for those 2 points. So at 10 seconds, x equals 10. Y, let's say about 7. And for 20 seconds, let's say that y is 14. So to find slope, remember slope is m, and it's change in y over change in x. Make sure you put your y's on top. So we could start with any order of our y's. I'm going to start with this y. So I'm going to say 14 minus the other y, 7, over. If I start with this y, I also have to start with that x on the bottom. So 20 minus 10. So we end up with 7 on top, 10 on the bottom, or 0.7. Our units are going to be these units divided by these units. So 0.7 moles per second is our rate on this example question. Just a few more things about enzymes that you should know. Let's review quickly. The substrate is the substance or substances that are acted on by an enzyme, and they bind like a lock and a key very specifically to a groove on the enzyme called the active site. When they get together, the enzyme has a slight change in shape called an induced fit or a conformational change. This improves the fit between the active site and the substrate and helps to catalyze the reaction, helps to make it happen and make it happen faster. Sort of a really smart sounding phrase that applies to enzymes is complementarity leads to a high degree of specificity. And what that means in this case is that the shape of the substrate and the shape of the active site, they have to be complementary. They have to fit together. And because of that, each enzyme can only catalyze one particular reaction because other substrates don't fit. Just the one specific to that enzyme fits. So again, each enzyme catalyzes one reaction. Most enzymes are found only in certain locations in your body. Now remember that enzymes are proteins. Proteins are coded for by your DNA. All the DNA in all the cells in your body is the same. That means that all your cells have the genes to code for all the body's enzymes. But you don't want all your body's enzymes to be in every cell. For example, you don't want digestive enzymes in your finger. That would be bad. So throughout your body, there's what we call selective gene expression. Certain genes, even though they're present in all parts of the body, are only working in specific places. And this is how different types of cells become specialized. And they do different things, and they look different, even though they have the same DNA. So another thing we want to talk about with enzymes are activators. These are substances which help to activate enzymes. And there's two main kinds we want to talk about: cofactors and coenzymes. Let's start with coenzymes. Coenzymes are usually organic compounds. They're usually made from carbon. Oftentimes we'll call these vitamins. So the main reason you need vitamins in your body is that they're substances that bind to specific spots on certain enzymes and help to activate them, help to cause them to work. For example, vitamin C helps to activate enzymes that make the substance called collagen, which is very important for holding your body together. It's one of the main components of your body's connective tissues. Vitamin A is a coenzyme for enzymes involved in sight. Vitamin D is a coenzyme for enzymes involved in making bone, for example. Without those vitamins, those enzymes don't work, and you get vitamin deficiency disease. Cofactors are very similar, except they're made from different things. They're made from minerals, usually some kind of ion, typically a metal ion. They have to bind to the enzyme, and again, they activate it. So without either a specific cofactor or coenzyme, it depends on which enzyme we're talking about, but without those cofactors or coenzymes, the enzymes don't activate, they don't work. So let's also talk about the effect of temperature on enzyme activity. And to do that, we're going to look at a graph. But first, let's mention the optimum or optimal temperature. There's a temperature that most enzymes work best at. And for most of the enzymes in humans, it's about 37 degrees C, which is pretty much the same thing as 98.6 degrees Fahrenheit. It's body temperature. There's a few exceptions to this rule, but typically, human enzymes have an optimum temperature of around 37 degrees C. So here's a graph, and notice that right about here, that temperature is around 37 degrees C. So that's the optimum temperature. That's the temperature that enzyme works best at. If the environment were cooled off, notice the rate of that enzyme catalyzed reaction slows down. This is not about denaturation. When things get colder, they move slower rather. Let me correct that. Because the substrates and the enzymes are moving slower, they're less likely to collide and therefore less likely to react. So coldness is not causing denaturation. But if the if the temperature of the environment exceeds the optimum temperature, notice the rate also slows down. Even though the enzymes and the substrates are moving faster, What ends up happening when you exceed the optimum temperature is that the shape of the enzyme starts to change, and that's called denaturation. Also notice that each enzyme has an optimum pH, and that optimum pH is a little bit above 7, just slightly above 7, for most enzymes in your body. And if the pH is changed to either a little bit more acidic or a little bit more basic, the enzyme starts to denature. Notice pepsin. This enzyme has an optimum pH of around 2. And that works for pepsin because pepsin is found in the stomach. If the pH gets more acidic than 2 and the pH drops, pepsin doesn't work as well. It starts to denature. If the pH goes above 2 and becomes more basic, pepsin doesn't work as well because it starts to denature. Trypsin is a little bit more representative of most of your enzymes in the body. Optimum pH a little bit above 7. It works best there. If the pH decreases and becomes more acidic, it doesn't work as well. It starts to denature. If the pH increases and becomes more basic, it doesn't work as well and it denatures. So this diagram is showing us maybe a little bit more what an enzyme actually looks like. Each one of these little blue circles represents an amino acid, because remember, enzymes are made out of proteins. Here we have the active site of our enzyme with a very specific shape and charge. And here we have the substrate fitting in there sort of like a lock and a key. The shapes have to be complementary. And remember that once it fits into that groove, the enzyme goes through a small change in shape, not a denaturation, just a small change. We call that the induced fit or conformational change. And that actually helps to catalyze the reaction to make it happen. So we also need to talk about inhibitors. Inhibitors are substances that help to stop an enzyme from working. Inhibit means to stop. So there's a couple of kind of inhibitors we'll mention. Let's first of all talk about competitive inhibitors. These are called competitive because they have a shape that's very similar to that of the substrate, and therefore somewhat complementary to the active site. So these are substances that are shaped like the substrate. They bind to the active site and they block it so the substrate can't get there. And therefore they prevent or at least slow down the rate of the reaction. An example is a type of medication called a sulfa drug. It's used to treat bacterial infections. Bacteria have this set of enzymes that convert a substrate called PABA to folic acid, an important vitamin, an important coenzyme for bacteria. We also need folic acid, but we get our folic acid from our diet. Bacteria manufacture their own. So what these sulfa drugs do is they inhibit the enzymes that make folic acid in bacteria. And if bacteria don't have folic acid, they die. These drugs don't hurt us because we don't have the enzymes to make folic acid. We take in our folic acid from our diets. There are also non-competitive inhibitors. These bind to an enzyme, but they bind somewhere other than the active site. They may bind where the cofactor goes, or the coenzyme. They may bind in an allosteric site, which we'll talk about in a minute. But the net effect is they still cause the enzyme to become deactive. They cause it to change shape and not work. Some examples are penicillin. Penicillin is a non-competitive inhibitor for enzymes in bacteria that create cell walls. So it helps to block the reproduction of bacteria and therefore causes them to die. Heavy metals, things like mercury, lead, cadmium, those all bind to enzymes and cause them not to work. And that's why these three materials are all very dangerous for humans to take in their body. They stop certain enzymes in your body from working, and if you take in enough of any of those substances, it can kill you. Arsenic and most poisons are poisonous because they're enzyme inhibitors. So something that's shown up on the AP exam the last few years are several questions about metabolic pathways. A metabolic pathway is a linked series of chemical reactions that occur within a cell. Usually the product of one step is the reactant for the next step. Some examples of metabolic pathways found in the body are the Krebs cycle, part of aerobic cell respiration, the Calvin cycle, part of photosynthesis. We might think of all of photosynthesis as a metabolic pathway. Digestion could be thought of as a metabolic pathway. Cellular respiration could be thought of as a metabolic pathway. We may talk about intermediates. So usually a metabolic pathway is this complicated series of reactions. You start with an initial reactant, you end up with a product. But in between those two, you make other things. The substances that are made between the initial reactants and the final products, those are called the intermediates. Since there's all these different reactions involved in a metabolic pathway, every step, every reaction requires a different enzyme to catalyze it and to make it happen. So a metabolic pathway is going to have a whole set of enzymes. Each enzyme, remember, is very specific and can only catalyze one reaction. Many of the metabolic pathways found in all living things are pretty much identical. For example, pretty much all of life does aerobic cell respiration the same way. The same chemical reactions, the same enzymes. Many of the digestive enzymes are shared between organisms. Photosynthesis works essentially the same way in pretty much all plants. Because they're the same enzymes, the same proteins, that also implies there's the same or very similar DNA. So because we share these metabolic pathways with other organisms, that implies common ancestry or evolution. So let's talk about feedback inhibition. Sometimes you'll hear this called allosteric inhibition. So here's a kind of a complicated sounding definition. The temporary deactivation of an enzyme brought about by an elevation of an end product of the pathway or the metabolic pathway. Feedback inhibition is a type of negative feedback. So you have all these metabolic pathways that make things in your body. Eventually you're going to get to a point where you have enough of that product, and it doesn't make sense to keep on making something that you already have a lot of, that the concentrations are already high inside the cell. So once the key word is concentration. Once a concentration reaches a certain point, the process is essentially turned off until some of that product is used up, and then the process is turned back on. So this is your cell being green. It's your cell conserving its resources, making what it needs when it needs it, and then when it gets enough, stop. Use some of that up. When the concentrations get low, start making it again. So let's look at a slide to show you how this works. So here's our initial reactant. Each one of these arrows here represents a different reaction. And here's what we're actually trying to make. This is the end product of the metabolic pathway. Notice that each reaction, each arrow, has a different enzyme, a specific enzyme, and that each step makes a certain metabolic intermediate. So let's say the cell has been running this metabolic pathway, and that the concentration of this end product is now high. There's a lot of it in the cell. Odds are that some of that product is going to bump in to the enzyme that catalyzed step one, or at least one of the early steps. It doesn't have to be the very first one, but usually it's one of the early steps in the pathway. That enzyme has two grooves on it. It has an active site where the substrate binds, but it also has another groove that we'll call either the allosteric site, or you'll see some books call it the feedback site. And what binds into that allosteric site is the end product of the metabolic pathway. And when it binds in there, it causes the shape of the enzyme to stop, to change. And therefore, this enzyme doesn't work anymore. Well, when this enzyme doesn't work anymore, its product isn't made anymore. Its product is the reactant for enzyme 2. So because this product isn't made, this enzyme doesn't work. This enzyme doesn't work. This enzyme doesn't work, and this enzyme doesn't work, which means no new product is made. So enzyme 1 is temporarily deactivated, and it tends to stay deactivated until the concentration of that end product drops. And once it drops past some critical level, the product that's sitting on this allosteric site will let go. Enzyme 1 will go back to its active shape. it'll start working again. And once it starts working again, it's making its product, which means enzyme 2 now has a substrate, so it starts working. When enzyme 2 starts working, enzyme 3 can work, 4 can work, 5 can work, and end products start being made again, and will continue to be made until the concentration reaches some critical high level. So feedback inhibition is when the product of a metabolic pathway actually turns off the pathway when the concentration of the product gets high. It's a temporary deactivation. It's your body using negative feedback to regulate levels of certain kinds of products. And that's it for enzymes.